Hello, class, and welcome to Introduction to Neuroscience. This week we will be exploring visual systems as well as auditory systems. Now, have you ever wondered how it is that we have developed our knowledge of the visual system? Well, it's an interesting journey of small little steps, which is indeed what typifies science. Small little discoveries that over time fill in the lattice, the picture of how a system works. Let's take a little bit of time now and just want to share with you a little story about how these discoveries occurred over a 100-year period to give us a good sense of the visual system. So back in the 1870s, an individual by the name of Franz Boll identified a substance in the frog retina that bleached when exposed to light. He termed it visual purple. But what he ended up discovering was something that we now know as rhodopsin, which is a key chemical protein in the functioning of rods in the eyes. Now, by the middle of the 20th century, it was well established that rods underlie night vision and cones underlie daytime vision. But how each cell was able to convert photons to electrochemical signals was unclear. Enter Selig Hecht and his team, who addressed this question of photoreceptor sensitivity in a clever and fairly groundbreaking study. In the 1940s, they directed flashes of light of varying luminescence into the eye of a subject who was sitting in complete darkness and measured detection performance. Now, by carefully accounting for the stimulus intensity and reflective and absorptive losses, they made an incredible discovery. A visual stimulus can be detected when it is comprised of as few as 5 photons. Now, remarkably, since their visual stimulus encompassed an area of around 500 rods, it was nearly impossible for any single rod to have absorbed more than 1 photon. However, what did the amplitude and kinetics of the photon-evoked signal look like, and how likely was it that a single photon response would occur? Well, that answer would have to wait for the development of what was known as patch clamp technology. Now, when that technology was finally developed, it was used by Denis Baylor and his team and applied to record rod photocurrents in the 1970s. Now, by recording from rod outer segments in toad retina, using suction electrodes while flashing a very dim spot of light, they measured responses exhibiting a binary amplitudes, which they defined as successes and failures, with the successes representing the single photon responses predicted by Hecht back in the 1940s. So, from the 1870s with Franz Boll through to Hecht's research in the 1940s, through to Baylor's research in the 1970s, we have 100 years of small little discoveries coming together to give us a picture of how part of the visual system works. I hope you enjoy this week. You take care. Bye-bye.